my area of interest has been in cancer, specifically in looking at the biology of cancer, the pathology of cancer. So this project that we've done on pancreatic cancer is consistent with that approach, where we look at the pathology of the tumours themselves, but now we're also able to look at the molecular biology of the tumours and the next generation sequencing, the very rapid ability to sequence the genome and RNA molecules, the transcriptome, and look at all the various changes that occur in tumours. The work that was published is a large-scale project which is under the International Cancer Genome Consortium, which is a number of countries, 22, have got together, and each country around the world has had decided to sequence the entire genomes of at least 500 cancers of a particular type. So in West Australia, we'd been working very much of establishing biobanks, and we'd set up the networks necessary to ask patients for their consent to go into theatre to collect the tumour material as it's taken out of a person during surgery, to work closely with pathology laboratories. And then the material that we collected was sent to the Garvan Institute for processing in Sydney, and the molecular sequencing was done through the Garvan Institute and the Institute for Molecular Bioscience at the University of Queensland. And the very interesting part of that is that we treat tumours, treat cancers, often as, or in the past, as one entity, as though there was such a thing called pancreatic cancer. But in fact, there were at least 4 subtypes that were defined through the molecular analysis. And one of them that was very unstable was associated with an inability of the genome to be edited and to be repaired. Ironically, that means that those tumours might be more susceptible to particular types of chemotherapy that target unstable genomes and target the ability to kill the cells that can't repair themselves readily. So it's one of those interesting things where the reason that you get the cancer also allows you to treat the cancer. And most recently, that work has been extended even further. And another Nature paper has just come out this week, which is talking about a further analysis of 457 of the tumours, and it further subgroups them into 4 types, very clearly telling us that when we do clinical trials in the future, we need to take very careful note of the tumour type and not treat them as one single homogeneous entity.